Hi, everyone. I'm very happy to be here. Such a beautiful space, and also I love this format of so many questions, so I'm hoping we can dig deep into ideas. So what I'm going to try to do today is to tell you about the POP-Z condensates, the bacterial condensates, and show you our data connecting structure and function. So we're actually going to look at individual kind of ultrastructure or individual components within the POP-Z condensate and try to connect structure and function. And for this audience, I'm going to show you a lot of kind of data and where we're at, but we don't actually have a theory that can really combine kind of the main message that I'm going to show you, that there's filament structure that drive function. So how can we get from these molecular filaments to properties such as surface tension or multi-phase behavior? We don't actually have the theory to do that yet, I mean, for us at least. So I'm very excited to be here with all of you guys and see if we can perhaps work together to come up with ideas of how to move forward with this. Okay, so to start, I have to introduce to you the system we're working with. And the condensate we work with is a bacterial condensate called POP-Z. It's part of, it's very important in the bacteria Colobacter crescentus. So I'm just going to give you 1 minute biology of Colobacter. Colobacter is a asymmetrically dividing bacteria. It has on one end this stalk here that is attached to a surface, and this cell will then grow to give rise to a daughter cell that is very, very different in terms of the transcriptional program and all of its behavior. And of course, also you can see this different appendage, which is a flagella. And upon division, the daughter cell will fly away, swim around, and eventually is going to differentiate back, differentiate into the stalk cell and give rise to another daughter cell. And this entire bacterium is only 2 micron long, but still is able to reproduce this behavior super reproducible, reliably. And how can it do that? We don't have membranes, okay. We can't really compartmentalize in the way that we thought about before. However, over the years, the community realized that there are different enzymes that are going to go to the two different poles of the cell. One set of enzymes is going to drive this, what we call this new pole behavior, and another set of enzymes is going to drive this old pole behavior. And for the sake of the talk today, we're going to focus on how they get there, or what brings them there, and what gets them there is this POP-Z protein. So at the two poles of the cell, we're going to there's this protein POP-Z that is, I'm going to show you now, forms a microdomain. And all of these enzymes are going into those by direct binding or indirect binding. Now, of course, there are two different sets, and it's the same protein, so how can it be? It's because, I'm not going to tell you today, but there are two other factors that are kind of like marking the identity. So the identity is not derived just by POP-Z, but by additional proteins. But for the sake of the talk today, we're going to stick with POP-Z. Okay, so what is this POP-Z protein? It's a simple scaffold protein, 177 amino acids. It has this N-terminal helical region where actually all the clients bind, and we know of at least 15 different proteins that bind in this specific region here in this helical regime. We have a disordered region that is very proline-rich and negatively charged, and if you actually compare this chemistry to the chemistry of IDRs in the human proteome, it's very different. So it's a very, very extended disordered region that is kind of unique, at least to what we see in human cells. And we have this helical C-terminal region that consists of 3 helices, and that's actually, I'm going to show you, going to drive condensation. If you get rid of POP-Z, you get a different, the cells is essentially very sick. It loses all of its asymmetry markers, can't do DNA replication segregation anymore. Cells are still alive, but they become this useless noodle in a way. Okay, So how is this POP-Z looking inside? So this is now tomography. These are actually all ribosomes that are all excluded from this microdomain. And there are not a lot of things, it's not, okay, I'm going to show you. It's not just large things that don't go in. First, I'm going to show you, these are single molecule tracks showing that molecules that can enter, that usually, again, have a binding affinity, are kind of, have increased dwell time within the condensate. So once you go in, you're kind of like in there for a while. But here I'm showing you actually just GFP molecules that cannot enter, they don't enter the condensate as well. And again, the selectivity of entry, at least from our experiment so far, is derived by the ability to bind. Okay, over the years we figured out what is the molecular grammar for this protein. So, as I told you, this helical region is recruiting client proteins. The disordered region is actually going to tune the material state of the condensate. And this oligomerization domain drive condensation. So we looked at POP-Z is found in alpha proteobacteria and only in alpha proteobacteria. And when we look at all of POP-Z within alpha proteobacteria, this is extremely, extremely conserved, like super. There are changes here, which probably because there are different clients for different microbes. But then for the disordered region, of course, we can align the sequence, but we found chemical features that are conserved. So we found that the percentage of proline, the percentage of negatively charged, and overall the length is conserved across different microbes. Question why? So what we did in this experiment is that we replaced the wild type POP-Z condensate with POP-Z that we just changed, we perturb these chemical properties of the disordered region. And what we've done then is on the x-axis here, we're measuring mobile fraction by FRAP. So to see, we're going to have some POP-Z condensates that are going to be more viscous and some that are going to be less. And on the y-axis, we're measuring growth by just serial dilution. And what we found was very interesting for us, and it is that there is kind of a Goldilocks effect. So if you're too liquid or too solid, basically the cells die. But if you're in the right regime of kind of viscoelasticity, the cells grow great. And I just want to give you one glimpse of what's actually happening, and we're going to look at the super liquids. Okay, what's happening to them? So here I'm just focusing on 3 of the perturbations that are looking at linker length. This is wild type, the extra liquid, which is just, we took the IDR and we just doubled it, literally, or made it like half the length. And now we're going to look at the serial dilution. So as I showed you before, that's wild type. That's when the linker is half the length, but that's when we make the linker double the length, basically the cells die. I mean, they just can't sustain it. Why is that the case? So if we're looking now at, where is POP-Z, that's where it is in wild type cells at the poles as expected, and also when the linker is half the length the same. But what's happening when it's super liquid, it actually cannot be maintained at the pole anymore, okay? That's a really interesting point. You see it just making these condensates outside from where it's supposed to be. And now we did like an extreme overexpression just to show you basically what's happening. This POP-Z liquid now is kind of taking all over the cell. Okay, and when we did tomography on this mutant, so that was wild type, where the POP-Z is nicely found in like where it's supposed to be, the ribosomes are away, it's all great, all good. But in this mutant, we're able to catch kind of like that type of cell. You see this POP-Z region in the middle, ribosomes are still excluded, but this thing is going to move around, okay? So there is this relationship between the different kind of condensates in the cell. And so I'm gonna go to this idea a little bit deeper, this is actually something I didn't show before. What actually maintains POP-Z at the pole, and why is it there? Turns out it's the nucleoid. Okay, the nucleoid is another condensate. It's actually the most important part of condensate in the cell. So here we took E. coli cells, and here we have POP-Z condensate and the nucleoid. And we know from many experiments in the past that POP-Z condensate will stay at the pole in E. coli if you express it. But what we've done is that we introduced a system that we added a system that can introduce DNA breaks, so kind of get rid of the nucleoid. So the question is, when we add arabinose and start getting rid of chopping up the nucleoid, what's going to happen? Is POP-Z going to stay nicely at the pole as a condensate, or just going to take over the cell? And what we found is the latter. So here I'm showing you POP-Z, DAP staining for the nucleoid, and the phase. And over time, as we do the treatment, the nucleoid is being broken and chopped, and then POP-Z, instead of staying as one nice condensate, is kind of starting to be diffused. And I'm going to show you just one. Oh, sorry. I don't know if I can do that. Okay, maybe not. But there are movies, really cool movies that show it. Okay, so I know I can try, but I don't know why it doesn't do that now. Sorry. Where's that? Oh my God. Okay. So I think the main point that I want to make here is that the material state of the condensate is not just, we know it's probably important for the function of the condensate too, but there are also the friends in the cell, okay? There are other condensates in the cell, and they kind of have to live together. And we have a lot of data that when we make POP-Z condensate to liquid, it actually don't have this separation of condensates anymore between the nucleoid and POP-Z, and actually things kind of mix, okay? And I think this is a really interesting concept, and it's probably also happening in the nucleus, where you have, I'm sure, different condensate, and perhaps these things matter too. Yeah, so this is something that we're still trying to figure out completely. But the fact that evolution kind of made sure that POP-Z condensates are in a specific set of material state perhaps also has to do with the environment of the cell, other condensate in the cell as well. Okay. So at this point, we knew a lot about kind of what POP-Z is doing in Colobacter, but we wanted to move more to the molecular level of trying to understand for each of these three models what they are doing. So that's an AlphaFold model of POP-Z, again with this helical organization domain, the disordered region, and the region where all the clients bind. And what we ask at this point is, how are we getting from a monomer to a condensate? And I really loved all the discussion that we had yesterday about the different clusters and how do they go into a condensate and what does it mean. So I'm going to show you kind of a structural biology perspective of going from a monomer to a condensate. And I think it will be really interesting to try and combine those ideas that we heard before to a system like that. So all of this work was spearheaded by Daniel School, a postdoc in the lab, was extremely productive and creative, and yeah, he's really great and moving all this project forward. So the first thing that Daniel figured out is actually for this POP-Z monomer to go into condensate, you need to add magnesium or low pH or high salt. So we have a temporal way to control condensation, okay, by adding one of those additives. So first we work in the dilute phase, and here we ran mass photometry, which is a way to get the oligomeric states of your complex. And we found that in the dilute phase, without addition of magnesium, we're getting trimers and hexamers. That's what the protein is doing. And this is now drawings that we've made with David Gutzel, trying to illustrate how those trimers and hexamers look like. So this is now the oligomerization domain creating this oligomerization domain creating this coil-coil region here with the disordered region kind of reaching out here, like the client, reaching out for the client. Like that. And that's the hexamer atomization of it. Really cool is that with the addition of magnesium still in the dilute phase, we're starting to get stacking of, we've seen multiples of 6. So we're seeing now species that are 12 mer and 18 mer. So again, with David, we kind of build a model of what we think is happening. We're perhaps getting stacking of these hexamers, one on top of each other. And then, so what I told you so far, yeah, we have the trimers and hexamers. Actually, with increased concentration, we can get to those filament structure, and with the addition of magnesium, we're going to get into condensates. There are really filaments in the condensates. So here we worked with Raphael Park, which is a lab next door to me at Scripps Research, and is a technician, Tamara. And here we took in vitro POPZ condensates, put them under cryo-ET, and you can actually appreciate these filaments, the structure here. And I'm going to show you the kind of like going under Z-stack, on Z-stack, to see this structure happening here. So that's the POPZ condensate in vitro. We also have pictures of POPZ in vivo. We can see the filaments, but it's a much more difficult system because there's a bunch of other stuff. So, they are there, but I can't tell you exactly the type of analysis that we're going to do here. Okay, so when we look at these filaments and try to, here we do a length distribution, we see that actually most of the filaments are shorter than longer, and I think for a lot of people in this audience that would be interesting to think about this isometric model that they fit and what does it mean exactly in terms of how can we combine that to the viscosity properties that we see for this condensate and surface tension properties. Okay, so we have a condensate made out of filaments. What's driving that filament structure? We know that condensation requires this helical region, but we wanted to know if this disordered region matters too. So what we've done is that we added a TEV site here in between those two parts. And before addition of TEV and no addition of magnesium, of course, we're going to see no condensation, as I told you before. But what we've noticed is that with the addition of the TEV protease, within 30 minutes, we're starting to see condensates form, okay? And if we spin it down, we see that the organization domain is corresponding to the condensates, and the supernate is everything else. And then Daniel, of course, went one step further and actually labeled with 2 cysteines here those 2 parts. And you can see that these condensates don't have the disordered region. In fact, they are devoid of the disordered region, and that those condensates are made out explicitly those oligomerization domain. We then went and did cryo-ET on these now oligomerization domain only condensates, and we again see those filaments, beautiful filaments, but you can see that we now have a very different kind of boundary. So there are differences in surface tension here. Okay, so basically the point is that all of the condensation is happening through this helical region. There's enough to build the filament, cross-link them, build the filament, cross-link the filaments. It's all done here. I just want to show you one mutant that we discovered. We found that the last amino acids were actually very positively charged. When we deleted those, we found something very interesting, which is we can still get condensation. However, these condensates are now made out of hexamers. So we now have a mutant that we can show that we can basically get condensation with hexamers instead of with filaments. And this is now the cryo-ET, so that's the POPZ condensate for the full length, but this is just now hexamers. And those are much faster, by FRAP. Okay, We're going to get to that mutant in a second. The last part that I want to show here on the molecular details is how is selectivity achieved. For that, we did now single molecule FRET with a Ashok-Denise lab. What you see here are the FRET signal, SM FRET signal for just POP-Z monomers. And you can see that there are two populations. One population with low FRET signal, meaning that those two points are far away from each other, and there's another population where they're close. So suggesting that the disordered region can kind of allow for this H1 to close back on this oligomerization domain. So actually, this is now how we see POP-Z in the dilute phase looking like, kind of like, we call it like an inactive hexamer, because as you can see in a couple of minutes. But when we add magnesium, we see a total shift. So, and with the addition of magnesium, all of that kind of high FRET signal is gone, and we just get this very, very, so it's all low FRET signals, suggesting that we get this conformational change. In the dilute phase, we have this like inactive condensates, and in the dense phase, we think inactive hexamers, and in the dense phase, we have this active hexamers. So why does it matter for the cell? We have those two conformations because all of these proteins, which we know a lot about actually, are found here in these condensates. Okay, when POP-Z, and it's very important actually for the cell function that they will be localized at the pole. We don't want this off-target binding. We don't want POP-Z to bind to clients outside of those regions. Otherwise, we might start to get clustering not at where we're supposed to be and not at the poles of the cell. So to kind of test this idea, we took one of those clients, ChIP-T, that is very important for cell cycle control. And what I'm going to show you now is that in the dilute phase, actually, ChIP-T doesn't bind POP-Z, which I'm showing you here through single molecule FRET, that addition of ChIP-T doesn't change the POP-Z FRET profile, and we showed it also through many other ways. So that's what's actually happening in the dilute phase. But in the dense phase, ChIP-T is totally inside the POP-Z condensate, 100%. And as a control, if we make those condensates that I showed you before with the TEV, that we have only oligomerization domain condensate that don't have the H1 where all the clients bind, ChIP-T is not entering. Okay, so yeah, so that's how this thing is looking in the dense phase. So we have now for POP-Z a conformational change that allow for clients to bind specifically in the dense phase, and that can be very useful actually for various POP-Z engineering projects that we do in the lab as well. Okay, so far I told you that the oligomerization domain is driving condensation by condensation of filaments. This disordered region is blocking this oligomerization interface in the dilute phase, and we have this phase-dependent binding. But what are the functional implications of maintaining a filament in the bacteria? So that's why I'm going to go back to that mutant that I told you about before. So this is what's happening in wild type. We get those hexamer that stack and get into this beautiful condensate. But we also have this mutant now, when we're losing the last 6 amino acids, that we can still get with addition of magnesium, different type of condensates. And if we were to express now, if we are replacing the wild type POP-Z with that mutant, the cell's dead, okay? No cells. I mean, no growth. Why? I mean, ChIP-T can still bind, like all the clients can still bind, but we have no, the cells can't basically survive. So for that, I need to tell you about one system in Colobacter, which is DNA replication, DNA segregation, that is happening through PAR-B. So PAR-ABS is actually a very important DNA segregation system in bacteria. Here you see PAR-B. It's binding to the origin, and it's basically creating these double condensates here. There's going to be a PAR-B condensate and a POB-Z condensate. And as the cell goes through its cell cycle, you're going to get DNA segregation. A new POB-Z is going to appear, and a new PAR-B is going to get to this new PAR. So what we did is that we just labeled PAR-B now in cells and looked at those two, how they behave as a function of the cell cycle. So here at the beginning of the cell cycle, those two are together. At 70 minutes, you get segregation of the origin to the other side as expected. So after 6 hours, it's all good, all the cells are growing. When you do the same now, but now, okay, we can't actually replace POB-Z with the delta 6, as I told you, because the cells are dying. So we just did a little bit of an overexpression of this mutant, this delta 6 mutant. When we do that, basically all DNA segregation is gone. PAR-B cannot find its place at the new pole, and it's just you can't do DNA segregation with a little bit of expression of this mutant. Now, you might think that, and also you can see that POB-Z is now not maintained at the pole anymore. That's actually just the delta 6 that is here. You might say that's because of that. So we've done a control where we express this delta 6, but with delta H1, meaning that the clients can bind. Okay, the clients can bind, but we are going to alter the ultrastructure of the condensate, which is the important thing. Still, we get no growth. Okay, so what delta 6 is doing is altering the ultrastructure of the POB-Z condensate in a way that doesn't allow for DNA segregation to happen. Okay, so last bit of data that I want to show, and this is again connecting structure to function. So this is the picture that we've worked on with David, basically showing you the POB-Z pole, and that's the POB-Z condensate. And here is the PAR-B, PAR-S, which is another condensate. And you see that they're beautifully separated. PAR-B binds both POB-Z and DNA. POB-Z cannot, is repelling DNA, but they have this beautiful kind of interface here. So what we've done is that we try to reconstitute this system in vitro. The question is, can we actually create the same thing that we see in the cell, but in vitro? Okay, perfect, actually. In vitro, and by doing that, maybe we can understand this structure function problem that we see. Okay, so we're going to have three components, which are going to be PAR-B, POB-Z, and DNA. So first, we have POB-Z condensates with PAR-B. Again, this is it. It binds it. And we see PAR-B going into POB-Z condensate, no problem, without PAR-S, so without the DNA. When we're adding DNA, we actually see this thing emerging where we see this multiphasic behavior where PAR-B is essentially sitting kind of at the tip where all the DNA, I don't have DNA labeled, but DNA is here outside. And we get that behavior as we see in the cell, which is very nice. But remember I told you about this delta 6. So delta 6 can still create condensates, right? And delta 6 also can still bind PAR-B. So now we have delta 6 condensates with PAR-B actually entering, it's all good. However, in this case, when we are adding PAR-S, so now we're adding DNA, something very different happens. Okay, we get a very, very different multiphasic behavior than what we got in the wild type. We're actually, POB-Z is now kind of like engulfing a PAR-B DNA condensate. And that's very interesting, again, because all the protein-protein interactions still happen. So PAR-B and DNA still bind, PAR-B and POB-Z still bind. But the ultrastructure of POB-Z changed in a way that now we get this very different multiphasic behavior. And what we think is that that's probably one of the reasons in the cell. So, why do you need the filamentous structure specifically? because you can make condensates in the, and why is it so conserved in alpha proteobacteria? We looked at this point 30 different POP-Zs from alpha. We purified them, and they all have like kind of the same structure. It's because you have to maintain this this ability to do the specific multiphasic behavior that you need. You need to make sure that you don't interact with the nucleoid and so on. So we're thinking that we are now starting to be able to connect those structural features to these like mesoscale properties. And again, we don't have kind of still the formalism to do that, but we have kind of all the knobs to play with that system. So to conclude, I told you that POP-Z forms liquid condensates, that the material properties of POP-Z are tuned to support optimal growth with this Goldilocks effect. Condensates are composed of filaments, and the IDR is modulating this behavior. And finally, this whole story that we're now starting to build up about this multiphasic behavior between the POP-Z condensate and the PAR-B condensate. And I would like to thank all the members in the lab, especially Daniel that drove a lot of this work, and now Anamika is starting to do a lot of the in vitro work as well, and collaborators and funding, and thank you for your attention.
Thank you. Questions? Yes.
So a question on the polar localization. So you mentioned one possible factor that's the change in viscosity. So the first question is how does change in viscosity affects localization?
Localization of what?
In addition, are there other factors that further help with polar localization?
Yeah. So you mean localization of the POP-Z in the
Yes.
cells?
Yes.
Yeah. So I think one of the beautiful thing about our system is it's modular, and we're able to change. So we are able to make alteration in the domain, in a domain that doesn't affect any of the binding interfaces. So the binding interfaces are maintained. So for your question, it's not that now you can't bind something that got you to localize to a specific place. It really is a kind of like a mesoscale property that is changing by us doing that. And it might be, I think the best example that we have now is these changes in these multiphasic behaviors that are happening because of changing in the structure.
Thank you.
Yeah, thanks. I'm very jealous that you've got David Goodsell illustrations all over. I was struck with the 50 millimolar magnesium for the filament formation, which my intuition, a little bit my experience suggests is a little high and can cause some changes. And the Goodsell diagram makes quite clear that the more biological thing is crowding. So I'm curious if you can get molecular crowding to drive filament formation.
We can get filamentation with 5 millimolar magnesium. We just use it for experiments. You actually don't need to add any crowder, even at 5 millimolar. You get condensation, we do have now data that's showing that the addition of the clients, I'm not showing you, but we have now new tomograms, actually with this PAR-B POP-Z, showing that there are changes in the structure, in the filament length and so on, with the addition of clients. So these are things that we will see,
Yeah, really beautiful. So at the last bit where you talked about like the different, like the mutant and the normal POP-Z interaction with the DNA and changes in the morphology, like how is it, what do you think is the relationship with the filament versus non-filament?
Yeah.
And maybe also like surface tension effects.
Exactly, I think it's actually surface tension effects because there's very different surface tension between when you get to the hexamer, you have a much higher surface tension. So I didn't show you that, but we have pictures of very different surface tensions between the two, and I think that that's what probably driving some of these multiphasic differences in multiphasic behavior. Yeah.
Hello. Very great talk. Like, if you look at the cryo-ET images, somehow it looks to me like the filament length on the interface is shorter than the filament in the center. Is there a correlation between, like,
it's
length?
a great question. And, you know, we heard so much about the interface, right, as being like its own environment kind of compared to the internal. We haven't done it here, but we are starting to look at that. It might be true. I think that they are definitely actually in a different conformation. I mean, they kind of look like in a different conformation to the rest of the cell. They have more, yeah, we can show you later pictures. But we are now doing more experiments, and actually we're doing single molecule tracking also to see if there are differences kind of in the dynamics at that interface compared to the middle. And with more and more analysis on the cryo-ET, we really have this opportunity now to see if there are different environments. Is it homogeneous, or there are actually different environments within the condensate? Yeah.
So I love this. I want to ask a very kind of strange question, which is, okay, you've got this system where the structured domains cause condensation, and the IDR either inhibits it or prevents it. That's not what most people think, right? When they look at these things, there are supposed to be these rules, and you've grown up in this world where, like, there's some. I mean, to call the paradigm, I think, is a little bit too much, right? But nonetheless, are these sort of heuristics that people use when they look at any particular protein, or they want to assign, right, the kind of how it's going to behave based on what the nature of the domain is. So. First, I'm just sort of interested in what your experience was like in trying to figure this stuff out. And second, do you have any insight into how it may be possible to look at a protein sequence, assign the domains, and then ask what their function is going to look like? Are there any principles that have come out as you've been on this little journey?
Yeah, I mean, that's a great question. This is not the only system that is actually having filaments of condensate. So the SPOP Montana, for example, is kind of working on similar principles. And there's also P62 that works on similar principles and so on. So there are now a bunch of, I mean, I think that as we kind of grow up as a community, we see that, yeah, maybe the first one we stumbled onto was an IDR driven. However, there are different ways to do it. And you know, we have, I think, now very kind of good ways to look for specific residues in the IDR that have higher valency and can drive condensation. We have it less for the structure domain, but I can say that we see it that kind of coil region are in all the examples that I've seen so far. It's like these are the kind of the features, but we don't have enough data yet to do it. Another thing that we kind of, a rule that we started to build that has to do with the condensates of filaments is that you basically need three functions. You need something to stack it, like you need something that will build the condensate. Very importantly, you need a capper, because you can't have the filament just grow forever, then you get amyloids, right? So you need a capper, and the last thing is that you need a crosslinker, something that will bring the filaments together. And if you look in literature, in other systems that have filaments, usually it's different proteins doing the three different functions, or at least two proteins. What's nice about our system, and I think because it's coming from bacteria, and bacteria just has to like have less resources in a way, is that all three functions are performed by the same domain. So I think that's kind of the power of this system, is that all three functions are by the same protein compared to the fact that you need the three different proteins. But this is how we're starting to think about it, like who is the capper, you know, who is the crosslinker, and to see it when we're looking at other systems too.
Yeah, that's really beautiful work. I have a sort of question, comment, suggestion following up on the crowding idea. I thought the data on the E. coli expression was very striking, where you digest the nucleoid.
Yeah.
But a slightly, possibly different wrinkle on that is that the nucleoid and the ribosomes are also phase separated, right? So if you digest, there could be an indirect mechanism as well.
Yes.
You direct the nucleoid, that gives more space to the ribosomes, less pressure on the POP-C. You clearly have thought of this.
Yeah, this is actually a really good point, and Alan's talk was really talking to that, but we try to like break polyzones too, to see if that's going to make the same. It doesn't do the same behavior, but I mean, I think there is so much to unpack here. And also when we're digesting the nucleotide, it's not that the DNA is gone, you know, it's just smaller. So we might get these like indirect effects. I think there's a lot more to do there. But yeah, this is the data, but I'm not sure it's 100% still on the interpretation. And I would love to talk to you more about that. Yeah.
Hi, Keren. Super cool system. I was just curious, you showed early in the talk this year, like really nice series of mutations that impact viscosity, maybe through the IDR, if I remember correctly.
Yeah.
Have you looked at those mutants and their impact on the multi-phase behavior? Like, is viscosity also important, or is it really just the surface tension of the filaments that's driving that?
Yeah, I mean, the experiment that we really need to do is to put all of them in our FRET system and basically see if this inhibitory effect is still happening. And because it might just be that you kind of need that. I mean, you saw we needed to add magnesium, right, to get condensation. That means that the IDR is playing an inhibitory role. But maybe when the linker is so long, for example, then the whole mechanism is different. Or maybe the other direction is easier. When you don't have the IDR is too short, you can actually do this inhibition. And that's why you get like too solid of a condensate. So I think it's more have to do with this mechanism of H1 kind of going to the oligomerization domain in the dilute phase and then opening up. So you kind of get like multivalency as a function of magnesium or client binding and so on. So we haven't done those experiments molecularly, but we are thinking about them, yeah. Thanks.
Any more questions from anyone? Yes.
Thanks. It's a really nice example of non-IDR driven condensation. I'm trying to understand still like what picture you have in mind though for how the IDR is shielding these oligomerization domains. Because with all those prolines, like I think they'll still be extended.
I think it all really boils down to basically those two conditions. As I told you, this oligomerization domain is doing the three functions. And the question is, when you have this inhibitory, like this inactive hexamer question, can you still form filaments? And second, maybe you can, but you can't cross-link them. So Daniel now is actually, I think he found like the mutants that will help distinguish filamentation from condensation. So now that we'll have mutants that can filament but can condense, we can really start answering these questions and try to understand where, you know, this inhibition, which step is it taking over? And that's kind of what we're trying to figure out now. Yeah.
Thanks.
Any other questions? Missing anyone? Yes.
So in the in Colvector was the double linker that moved away from the pole, and in coli where you expressed it and it moved. So how mobile is that big condensate? How does that happen? Because if you got even there, you've got the nucleoid in Colvector that's still there. And so does it break up into small things and go around the nucleoid, or it just like charges right through, or have you double labeled DNA?
Yeah, so we didn't. Right, you're right. So I mean, we didn't. They kind of I've done these experiments where I have DAPI stain, and you kind of see POP-Z going like across, like. They're still separated, but they are not. Yeah, it's kind of where the surface. But it's so. I mean, it is strange. But yeah, in movies, within seconds you see it kind of. It can take the cell within 30 seconds, all over. It's very. Yeah, I mean, it is strange. But I think the important point is that evolution actually made sure that that condensate would not be too liquid, and then you can start to think about, is that a general principle? You know. Our bacterial condensates always have to keep viscosity at a certain regime to do that.
If you wanted to localize the pole to somewhere else.
But still, like maybe it cannot be maintained. I don't know, unless it's maybe binding to DNA. But if we think of like all the others that we know of, like those RNA condensates and so on, like maybe they also cannot be two liquid for a reason.
Lovely talk. A question on the two poles. You mentioned that they form on both, but there's some markers that distinguish the new pole versus the old pole. We should not think too much about it.
No, we know. I just didn't talk about it. I can talk about it for an hour. Yeah. Okay.
Are they just markers that one think of that distinguish old state and new state, or are these two functionally different micro compartments?
Yeah. Okay. Yeah. So the cool thing is that they are functionally two different. What's happening is that you get the new pole that get one marker, and then during differentiation, when like the swarmer is transitioning to be a stalk, that marker is actually being degraded. Okay, the cell is working really hard to get rid of it, and a new marker is transcriptionally is being transcribed at the right time to replace that one, come in, and because of that different combination of A and B compared to A and C, you get a totally different composition. So to make sure that identity is changed at the right time is a whole cell cycle, a regulated process. Yeah.
Okay, with that, let's thank our speaker again.